Cells, if you recall from the cell theory, are in fact the basic unit of life. However, few cells can actually be seen with the unaided eye. This diagram represents a scale of visibility that shows you what you can actually see with the human eye as opposed to the microscope. With the human eye, you can see a variety of things. You can see things that are very large. For instance, a giant sequoia tree, elephants, humans, actually a single cell, a hen egg. However, with a microscope, you're actually able to see significantly more than you can with the unaided eye. With the microscope, for instance, you could see life, single-celled life, in pond water, such as the paramecium. You can also see human eggs, human red blood cells, and bacteria. Another type of microscope, the electron microscope, that actually uses an electron beam to actually illustrate or identify structures, can be used to see even smaller particles, such as viruses, or organelles like ribosomes, proteins, amino acids, and even actual individual atoms. Cells range in size from a few micrometers to several centimeters. And it is advantageous to have a large surface to volume ratio. If you go ahead and you look at these two cells right here, to the right, they're very different sizes. One is very small and one is a lot larger, and they have drastically different surface to volume ratios. In the case of the cell on the left, what you see here is a cell that is going to go ahead and have a greater surface to volume ratio than the cell on the right. As cell size increases, such as you see with the cell on the right, volume grows much faster than surface area. Most cells are small because larger cells do not function as efficiently. If a cell has a larger surface to volume ratio, that means it can get more of the important stuff that needs to come into the cell into it, as well as stuff that needs to leave the cell out of it. Stuff that needs to come into the cell will include nutrients, as well as oxygen to go ahead and create energy, and stuff that needs to leave the cell will include wastes, as well as carbon dioxide that is created in the process of energy production. A cell's surface provides the interior's only opportunity to interact with the environment. The more opportunity a cell has to interact with the environment, the better. So hence, a large surface to volume ratio is very important. Cellular structure is organized. First of all, for any given cell, you have a plasma membrane encasing it or surrounding it. The plasma membrane represents a cell boundary that controls permeability of the cell to water and dissolved substances, meaning that the plasma membrane will control flow of things into and out of the cell. Inside of the cell, what you have is a fluid matrix called cytoplasm. It fills the interior of the cell, and within the cytoplasm, you're going to have several things floating within it, such as organelles. In this diagram, you can go ahead and see where the cytoplasm is indicated in the orange color, and surrounding that is highlighted in purple, you have the plasma membrane. Plasma membranes have a polar region as well as a nonpolar region. The polar region is going to go ahead and be composed of a phosphate chemical group, while the nonpolar region is going to go ahead and be composed of fatty acids. If you look at the diagram on the right, you're going to go ahead and see the structure of the plasma membrane. The plasma membrane is actually composed of molecules called phospholipids. And so you can see the polar region as well as the nonpolar region. The polar region is actually going to be a type of region that is water soluble, while the nonpolar region is water insoluble. What this means is that the polar region is going to go ahead and be touching areas on the inside and the outside of the cell. It's going to go ahead and be touching those areas that are water-based, whereas the inside or the interior of the plasma membrane is going to go ahead and be nonpolar. It is going to go ahead and not be touching areas that are water soluble. The basic structure of the plasma membrane is represented by something called the fluid mosaic model. The plasma membrane is represented by a phospholipid bilayer with embedded proteins within it. So what we see here on the right-hand side is the structure of the phospholipid. This represents one phospholipid, a polar region and a nonpolar region, a polar head and a nonpolar tail. The polar region is also referred to as hydrophilic or water-loving, while the nonpolar region is hydrophobic or something that avoids water. Keep in mind what we have is a phospholipid bilayer, so hence we're going to go ahead and have two phospholipids, whereby you're going to go ahead and have a polar region, and coming from that the nonpolar region, and it's going to go ahead and be connected to another phospholipid molecule. In water, phospholipids spontaneously form a bilayer, and as mentioned, the plasma membrane is going to be composed of two phospholipid layers, or it's going to go ahead and be composed of a phospholipid bilayer. What you see here in this figure are two phospholipid layers. Number one, the first layer is going to go ahead and be composed of, again, those hydrophilic heads or water-loving heads and the hydrophobic tails. And adjoining to this hydrophobic tails will be the hydrophobic tails of the second layer of phospholipids, and below that the polar hydrophilic heads. What you essentially have here, again, is this plasma membrane composed of these two distinct layers of phospholipids. The outsides of the plasma membrane, where you have your polar hydrophilic heads, are going to go ahead and be touching the outside of the cell, which is water-based, as well as the inside of the cell, which is going to go ahead and be water-based and will consist of cytoplasm. The hydrophilic heads are in contact with aqueous layers, and the hydrophobic tails on the interior of the bilayer are going to go ahead and be found away from water. There are several proteins within the membrane itself. These are going to go ahead and be considered membrane proteins. They're going to go ahead and be found within and on the plasma membrane. You have two different types of membrane proteins. Number one, cell surface proteins, and number two, transmembrane proteins. Cell surface proteins will go ahead and project from the surface of the membrane, and these cell surface proteins will often act as markers or receptors of a given cell. Transmembrane proteins will extend all the way across or through the bilayer, hence they are transmembrane. They are crossing the membrane. And they are actually going to go ahead and provide channels into and out of the cell. This diagram indicates for you proteins that are embedded within the phospholipid bilayer of the plasma membrane. The plasma membrane itself is indicated in white. Notice that you have two phospholipid bilayers. Remember, each phospholipid bilayer is going to go ahead and be composed of hydrophilic heads and hydrophobic tails. The hydrophilic heads are going to go ahead and be indicated by the white circles. The white plasma membrane is going to be bound on either side by an area that is aqueous. Above the plasma membrane, you're going to go ahead and have the outside of the cell, while below the plasma membrane in this particular diagram, you're going to go ahead and have the inside of the cell, which is going to be represented by cytoplasm. In purple, you have your cell surface proteins and your transmembrane proteins. Notice that for your cell surface proteins, you have a series of globular proteins that are going to go ahead and have emanating from them carbohydrate chains. These carbohydrate chains are sticking outside of the cell surface, and they're going to go ahead and indicate to other structures outside of the cell surface what this particular type of cell is and what specifically it does. You also have your transmembrane proteins, and these transmembrane proteins are going to allow passage of structures from the outside of the cell to the inside of the cell and vice versa. There are two major kinds of cells. Number one, we have prokaryotic cells, also referred to as prokaryotes. and also eukaryotes, or your eukaryotic cells. Your prokaryotes appeared about 3 billion years ago, and they are going to go ahead and be your bacteria and your archaea. There are over 5,000 species that are currently recognized. And these prokaryotes that consist of both bacteria and archaea come in 3 main shapes. Number 1, you're going to have rod-shaped bacteria. Number 2, spiral-shaped bacteria. And number 3, spherical-shaped bacteria. The first group of cells, the prokaryotes, have a very simple architecture. And they have a very simple architecture because they're going to go ahead and be far older than the eukaryotes. These prokaryotes are going to go ahead and lack a nucleus, which is going to go ahead and be a membrane-bound structure that contains the DNA. In addition, they are also going to go ahead and lack other complex membranous organelles. The diagram that you see here represents an example of a typical prokaryote. The structures indicated in blue are going to go ahead and be found in all prokaryotes. Number 1, what you're going to go ahead and find is a plasma membrane, and within that plasma membrane, you're going to go ahead and find cytoplasm. You also are going to go ahead and have DNA. However, again, keep in mind that that DNA is going to go ahead and be found floating in the cytoplasm and not found within a nucleus like you would find in eukaryotes. Instead, it is found in what's called the nucleoid region. And notice that you have one single chromosome rather than several chromosomes. You also are going to have several ribosomes that are floating within the cytoplasm. And these ribosomes are extremely important. They're important specifically for protein synthesis. There are several other structures that you might find in prokaryotes, including the following. Number 1, you might also have a cell wall that is going to be surrounding the plasma membrane. You additionally could potentially find something called a capsule. And the cell wall and the capsule would provide additional structure to the prokaryote. You also might have a flagellum. This is going to be a whip-like structure that extends from the cell. And the flagellum would be important, number 1, for movement or locomotion, or also for feeding. And finally, you're also going to go ahead and have something called a pilus. Now this specific structure is going to be for attachment, or also could be used for genetic exchange. Your eukaryotic cells, on the other hand, appeared about 1.5 billion years ago. Hence, they're significantly younger than your prokaryotic cells. Your eukaryotic cells are going to go ahead and include the following kingdoms: Protista, fungi, plantae, and animalia. And this diagram to the right, this phylogenetic diagram that illustrates for you relationships among organisms, shows you in red that you have your prokaryotes, eubacteria and archaebacteria, and you have your eukaryotes separated from them. Again, your animalia, your plantae, your protista, and your fungi. Your eukaryotic cells are going to go ahead and be larger than your prokaryotic cells, and your eukaryotic cells are also going to be far more complex. Number 1, your eukaryotic cells are going to go ahead and possess a true nucleus. And this nucleus is going to go ahead and house genetic information, or DNA. In addition, your eukaryotic cells are also going to go ahead and contain membranous organelles, or organelles surrounded by 2 membranes. And finally, they also will contain something called the cytoskeleton, which will go ahead and provide structure to a given cell. This diagram represents for us a typical plant cell. Let's go ahead and look at the components of this plant cell. If we start at the inside of the plant cell, we're going to go ahead and see in purple, or number 5, the nucleus. The nucleus is going to go ahead and house the DNA, or the genetic information of the cell. The nucleus is going to be surrounded by the endoplasmic reticulum, or the ER, which is going to be indicated by number 6. And this structure is going to go ahead and be responsible for the production of macromolecules. Also, as part of number 6, you're going to see the Golgi complex. And the Golgi complex will go ahead and transport things that are produced by the endoplasmic reticulum. Other structures that you see in this particular plant cell include the following. Number 1, you're going to see number 4, the cytoskeleton. These structures are going to go ahead and provide structure to the cell and also play a role with regard to movement of things inside of the cell. If you go ahead and look at number 3, you're also going to see mitochondria. These mitochondria are important for the production of energy. And if you look at number 9, you're going to go ahead and see chloroplasts. These things are going to go ahead and harness the energy from the sun and turn them into food for the cell, or nutrients for the cell that ultimately can then be transformed into energy. The large blue structure in the middle of the cell, number 10, is referred to as the central vacuole. This central vacuole can go ahead and allow water to enter the cell or leave the cell. It's basically a storage point for the cell, and it can allow the cell to increase or decrease in size. And if you look at number 8, that is going to be the cell wall. The cell wall is going to go ahead and surround the plasma membrane and provide additional support specifically to plant cells. Those structures indicated in the red box, number 1, chloroplasts, number 2, the cell wall, and number 3, the central vacuole, are going to be found specifically in plant cells as opposed to animal cells. Let's go ahead and take a look at a typical animal cell now. In this particular case, let's start from the inside out. Again, what you find here in purple is the nucleus in the animal cell, just like you found in the plant cell. And again, this nucleus is going to house the genetic information. The nucleus, again, is surrounded by the endoplasmic reticulum and the Golgi complex, indicated by number 6 in this diagram. Other important elements are going to include the number 1, the mitochondrion, indicated by number 2 in this particular diagram, and also the cytoskeleton, indicated by number 4 in this particular diagram. I also want you to notice number 12, the centrioles. The centrioles are going to represent a complex assembly of microtubules that occur in pairs and are going to be important for division of the cells as well as for movement within the cells. Now, what you have here is a situation where you're going to go ahead and have several things in common with a plant cell, but several things being different. Notice that, of course, in this particular animal cell, you're going to also have a plasma membrane. But unlike the plant cell, you do not have a cell wall in this particular case. You also don't have that vacuole that we discussed previously. And we also do not have chloroplasts. But we do have these centrioles, and the centrioles are not found in plant cells.